Welcome everyone to Iris Biotech's 15th online workshop. True to our slogan, Empowering Peptide Innovation, we're keen on knowledge exchange and getting in contact with scientists all over the world. Therefore, we started this workshop series 3 years ago. Today, we deviate somewhat from pure peptide synthesis chemistry and look at a novel method of bioconjugation, which is, of course, also suitable for peptides. Our guest speaker, Professor Maleimide from Universiteit Ghent, will present fascinating results of her group's latest research to the construction of furan-derived building blocks that can be used as a new conjugation moiety. My name is Raimund Meyer. I'm working as COO at Iris Biotech, and today I will guide you through this workshop. As always, I would like to begin with some technical information. To ensure good data transfer of the presentation, all participants are automatically muted and your video cameras are switched off. This live event is recorded, thus if you enter late or miss anything, please check on the bottom left side of your Teams screen if you are watching the recording or if you are live, which is shown by the red broadcasting symbol. You have the possibility to pause the video, rewind or jump back to the live session. In addition, the recording of the workshop will be uploaded on our homepage afterwards. Questions can be submitted by using the Q&A chat of Microsoft Teams. You can enter your name or submit your questions anonymously. Initially, your questions are just visible for the organizers and will then be published to the audience, so don't worry if your submitted questions do not appear on the chat screen immediately. If you have any technical queries, you can also submit them in this Q&A chat. Our technical assistant will then connect with you and try to solve the problem. I would now like to give you an overview on today's agenda. We'll start with a short company introduction by our Chief Marketing and Sales Officer, Karin Rustler. And afterwards, our external speaker, Professor Maleimide, will present to you the results of her group's journey from furan and singlet oxygen to clever, innovative Maleimide replacement technology for bioconjugation. As mentioned, questions will be collected throughout all talks in the Q&A chat, but of course, you can also get in contact via email or phone call after the workshop. Let's now start with a short introduction to our company by my colleague Karin Rustler. Karin obtained her doctoral degree in organic chemistry at the University of Regensburg. Beginning of 2020, she joined Iris Biotech as a project manager, and nowadays she's our Chief Marketing and Sales Officer. Karin, thank you for your time. Please go ahead with your presentation.
Thank you, Raimund, for the kind introduction. And also welcome from my side to today's workshop. As Raimund mentioned, I will start with a brief company introduction for those of you who do not yet know Iris Biotech. But don't worry, I will keep it really short before we continue with our highlight today. For this slide, I just want to focus on a few aspects. The company Iris Biotech has been founded in 2001. Our portfolio comprises more than 6,000 different products. Of course, we cannot provide or produce all of them in our own synthesis laboratory in Willstedt, but we are working together with different manufacturing partners around the globe to realize demands from milligram up to kilogram quantities for research purposes up to GMP quality. To give you not only facts and figures, I always like to share the picture of the beautiful landscapes we are surrounded by, and I can highly recommend visiting the beautiful so-called Fichtelgebirge, especially also during winter season for sleigh riding. On this slide, you see a picture of our company building where we are operating from and where our orders are being processed. As mentioned briefly in the beginning, on the one hand, our mission is to support our customers in their ongoing projects from research scale to commercial production. In this regard, we are mainly active in the field of peptide synthesis and modification by providing amino acids, resins, reagents, and solvents, but also in related fields such as linker technologies and products for drug delivery. But for us, our slogan, Empowering Peptide Innovation, does not only mean to provide innovative products, but also to act as scientific advisor by providing and exchanging knowledge. In this regard, we build up our knowledge base, which includes topic-specific brochures and flyers, which can be downloaded from our website and which are updated on a yearly basis. Besides, we are publishing a bi-weekly newsletter. If you are not yet registered, please refer to the link shown on the top right side. And yeah, with this, I would already like to thank you for your kind introduction, for the kind attention, and also the introduction by Raimund, and pass on to our guest speaker.
Yeah, thank you, Karin, for giving this introduction. Yeah, let's now really go on with our highlight and switch to our guest speaker, Professor Annemike Madder. Annemike obtained her PhD in organic chemistry from University Ghent in 1997. After that, she went for postdoctoral studies to the University of Milan and to the Karolinska Institute in Sweden. She then started her independent career returning to Ghent to make up tenure as assistant professor in 2002. She was promoted professor at the Department of Organic and Macromolecular Chemistry in 2014 and full professor in 2022. She currently is heading the Organic and Biomimetic Chemistry Research Group, specialized in the design and synthesis of modified peptides and nucleic acids and methods for their conjugation and labeling. She is currently a member of the IOF, the Industrial Research Fund Council, and serves as substitute member in the Special Research Fund Council at the University of Ghent. From 2010 until 2015, she was a member of the ERC Starting Grant Panel, which she was also invited to chair in the years 2013 and 2015. Annemike, it's a great honor to host you as our guest speaker. The stage is yours. We're looking forward to hearing about your research. And for the audience, please post your questions by using the Q&A chat.
Okay. I suppose you can hear me now well? Yeah. Okay, thank you. So thank you, Raimund and Karin, for this very kind introduction. And I would like to thank also Iris Biotech and more specifically Thomas Brückdorfer for being very encouraging for this kind invitation and also for really sticking to the mission statement that you just said and really supporting us as researchers in the translation from research to final applications. And so with that, I really want to introduce you to the methodologies that we developed within our group, where we use Furan as a tool to achieve cross-linking and also to construct building blocks that can serve as Maleimide alternatives. And so, first of all, I would like to tell you something about Furan itself and why we use that small heterocycle. Yeah, because as such, as an aromatic moiety, this is rather stable, and you would not expect this to be useful for bioconjugation reactions. However, it is known that furan can be specifically oxidized to this reactive moiety. And so when you insert it in a biomolecule, you can benefit from that same scenario to use the furan as a kind of gauged electrophile. Yeah. By achieving that oxidation at a very specific moment in time, you can generate reactivity on demand, which means that you can avoid collateral damage and have spatial temporal control over your crosslink reaction. And so just to set the stage, I want to introduce you to our, what I would call OBCR's Furan technology platform. It all started when we were starting by introducing furan into an oligonucleotide, pairing it up with its complement, and then looking at that crosslink reaction. We later on translated that to the peptide and protein applications, where we look at peptides that contain furans to then crosslink to nucleophiles, but I'll go more in detail afterwards. And last but not least, I want to introduce you to our new bioconjugation tools that also come from that same furan through an oxidation reaction, generating these what I would call Maleimide alternatives. To set the stage, I would like to tell you how we actually developed that chemistry. And so you can see here that we started by incorporating a furan modified nucleoside into a natural oligonucleotide, which is then paired up with its natural complement, as you can see here. The furan now faces a residue on the other side of the strand, in this case a C. Upon oxidation of the furan moiety, and this can be achieved by treatment with N-bromosuccinimide or singlet oxygen, which can be generated via irradiation of a photosensitizer, the furan is then transformed into this very reactive enal moiety, which then reacts with the opposite strand, a nucleophile on the opposite strand. In this specific case, in a nucleic acid scenario, it is the exocyclic amine attacking the aldehyde, leading to this adduct, which is then cyclizing and rearranging to this adduct, which can further dehydrate to a stable crosslink. So you can see that in this case, furan as a small unreactive moiety can be triggered so that we can induce that reactivity. And so what we learned in this whole nucleic acid scenario is that it is a very proximity sensitive reaction. So we really need the nucleophile on the other strand, close by to the oxidized furan, which allows us to avoid unspecific and off-target reactions. The methodology in nucleic acids has been published and in the current protocols in nucleic acid chemistry, but this is just to give you the background. Yeah. Today we will more focus on the peptide and protein scenarios, where we use that same furan in proximity to a protein, and then we need a protein nucleophile in close proximity. So how to incorporate a furan moiety in peptides? And so this is rather easy, and also Iris Biotech makes that rather easy because there's a commercially available derivative of furylalanine, namely the FMOC2 furylalanine, which can be incorporated in a peptide sequence very easily by solid phase peptide synthesis. Given the fact that this amino acid is isosteric with histidine and isoelectronic with tyrosine, it has a minimal influence on peptide structure and electronics, as I will show you later on. So the first scenario in which we applied this furan modified peptides was in a setting where we try to crosslink in vitro the furan modified peptide to a protein. And you can see the sequence or the complex that we used for that. In green, you can see the actin protein, and in red, you see tyrosine beta 4, which is a ligand actually of that actin protein and forms a tight complex with the actin. So in that complex, we can now introduce, or in the tyrosine beta 4, we can introduce the furylalanine by solid phase peptide synthesis. And when you look at the crystal structure, you can see a side chain of a lysine indicated here, which is in close proximity to that furan. The furan can then be oxidized, and for that we use a photosensitizer, such as either methylene blue, rose bengal, or TAMRA, which is then irradiated and can convert triplet oxygen into singlet oxygen, which then oxidizes the furan into the keto-enal, which is then the reactive moiety, and upon close proximity with an attacking nucleophile, we can then generate a complex that you can see here and form a stable keto-amide linkage. Analysis by Western blotting, you can show here that this is the original protein, this is the cross-linked complex actin-tyrosine, and so we can then isolate that cross-linked protein and identify the cross-linked residues. And we were able to show that this is indeed the bond which is being formed between the furan and the lysine, which is proximate in that complex. So we then went for furan peptide probes for GPCR target identification and tried to do this on live cells. In general, you might know that ligand GPCR interactions are very important, and that so a lot of drug design is based on these ligand GPCR interactions. So making ligands that can cross-link to their target GPCR can be used for target identification, or can also be used to construct ligands for imaging so that you can localize your GPCRs on cell membranes. You see that we have been studying, or I depict here the 3 cases that we have been studying. And for the purpose of today, I will focus on that first system here, the Kispeptin-10 GPR54 system. The Kispeptins are endogenous peptides of which various forms exist. And we were able to do this study with the shortest analog of that, which is the 10-mer. A crystal structure is available where you can see this peptide here with a tryptophan moiety in the active site. And zooming in on that active site, which we will do in a minute, shows us that there are various nucleophiles around. In this case, the therapeutic relevance of these peptides is that they can be regulators of reproductive maturation and function, and that they are used in control of fertility. You see the detailed structure of the KP10 here, the endogenous Kispeptin-10, which is depicted here. And so if we look at the crystal structure, which was done together with University of Orleans by homology modeling, you can see that the tryptophan moiety of the KP10 is surrounded by nucleophiles of the GPR54, giving us confidence that maybe this scenario, replacing the tryptophan by a furan, will work and we can achieve cross-linking in that way. So we constructed a few peptides, and so you can see the wild-type peptide here, which we modified with a biotin to later on allow visualization of the cross-link reaction. And then we synthesized the peptide where the tryptophan was replaced by the furan. We also synthesized a scrambled peptide where the furan is introduced in a similar position, but the rest of the peptide is scrambled. And so, as you can see in the next slide, the calcium mobilization assay, which is used here to check whether these peptides are still biologically active, this indeed learns us that the blue curve here for the human KP10, then we can see that the biotin PEC human KP10 is even a little bit more active, and also the furan modified analog, the black line, is equally active. So we don't lose activity by substituting the tryptophan for phenylalanine. On the other hand, the scrambled one, which contains the furan, but for the rest, the sequence is scrambled, has, as expected, no biological activity. Knowing that the biological activity was still okay, we can use these furan-modified peptides to, in a solution, and incubate living cells with this peptide. And so, upon interaction of the biotin furan Kispeptin-10 with the GPR54, furan oxidation then leads to this enal and the cross-linking. And you can see here that a Western blotting analysis, this is just a control. So the receptor is visualized by these green spots, where we have an intracellular and an extracellular GPR54 fraction. These red spots come from cellular biotinylated proteins. So this is just a control on Western blotting. Then when we incubate the cells with the furan-modified peptide, we can see here that the extracellular fraction of the GPR54 now turns from green to red and yellow, which means that here we really have the protein, which is now covalently linked to the furan peptide. If we do the same thing with the wild type or the scrambled peptide, we don't see the cross-linking here, which means that we can actually use these peptide ligands incorporating furan for targeting cell surface receptors, GPCRs, and achieve cross-linking. Most interestingly, in this scenario on live cells, we do not need external triggers. The furan can be oxidized by reactive oxygen species that are generated on the cell membrane by NOx enzymes. So upon incubation of the live cells with this furan-modified peptide, we can benefit from spontaneous furan activation by endogenous ROS species. No need for externally adding oxidizing agents. And so these furan peptide ligands can be used for target identification. and pull down. This furan oxidation scenario actually inspired us to do the following. So here you see the oxidation reaction which we have used until now to the diol. In collaboration with the laboratory of Professor Georgios Vasilikogiannakis at the University of Crete, the following scenario was investigated. If we incubate that with an external amine, then these two hydroxypyrrole building blocks can be formed, which can be further treated with methylene blue to achieve these 5-hydroxypyrrolidine 2-ones, in short, 5-HP2O building blocks, which in our minds were very similar to maleimides. Now, maleimides are often used, as you might know, and as also Iris Biotech knows, for protein modification. And in a protein, they specifically target cysteine. Cysteine is the amino acid residue with a thiol in the side chain, which has a very low abundance and is a highly targeted amino acid. It can also easily be introduced by mutagenesis. On top of that, it displays a unique nucleophilicity with respect to all the other nucleophiles in the proteins. So that's why the combination of a thiol or cysteine-containing protein with maleimides is often exploited in the following scenario. The protein with a thiol can be used to do a Michael addition on that maleimide, and then once this conjugate is formed, hydrolysis leads to an irreversible reaction, and you can have the final conjugate. Of course, this hydrolysis, depending on how it occurs, can generate a little bit of a heterogeneity in your sample. Also, what you see here is that this reaction is reversible. And these are some of the problems that you can notice when using maleimides. First of all, the building blocks are very sensitive to hydrolysis. And whereas the hydrolysis is a desired reaction at this stage, it is a non-desired reaction at this stage, because hydrolysis of the maleimide building block will lead to a species that is not prone to bioconjugation anymore. When we form the conjugate, and before the hydrolysis, because of the reversibility of this Michael reaction, retro-Michael reaction can take place. And in the presence of a large excess of other thiols, we can have a thiol exchange reaction, which actually leads to the loss of payload. And given the high amounts of glutathione or BSA in vivo environments, this is a frequently observed problem. And so, given these limitations of hydrolysis, retro-Michael reaction, and thiol exchange, the search for new cysteine selective conjugation crosslinkers is still ongoing, as you might notice from literature. So we set out to investigate the potential of our 5-HP2O building blocks for bioconjugation with cysteine. And so the first thing we checked is, what is the hydrolytic stability of these building blocks? And as you can see here, under a variety of conditions and over prolonged periods, these building blocks are very stable. You can see here that this is just a dummy one where we have, let's say, an innocent side chain on that molecule. If we compare that with how maleimides behave in these at pH 7.5, for example, you can see here, this is our 5-HP2O building block at pH 7.5. After 5 days, still no sign of hydrolysis. If we look at the corresponding maleimide, you can see that even after a very short time, you can see next to the original maleimide, you can see formation of the hydrolyzed one. And after 5 days, nothing is left of the original maleimide anymore. And you can see that even after 1 hour, you have a 50-50 ratio of the active maleimide, but also 50% of that non-reactive building block. So hydrolytic stability of our 5-HP2O building blocks is definitely much better. Then let's look at the conjugation reaction. And so we originally thought, if we look at the maleimide, the thiol reacts in a Michael addition fashion, and we expected a similar thing on the 5-HP2O building blocks. Now, great was our surprise when the observed structures by NMR showed the thiol to be located on the carbon next to the carbonyl, and not this one here, which we actually anticipated to be attacked in this fashion. That led to a series of investigations where we could then confirm this mode of conjugation, where we can say, okay, this 5-HB2O is actually opening up. The thiol is now has the choice to attack in a 1,4 addition mode, either to the ketone or to the amide. And then the most reactive position is this one here, where we attack in the unsaturated position with respect to the ketone. So that leads to the formation of this carbon-sulfur bond, after which the 5-HB2O reforms through cyclization. So, and this is how the sulfur actually ends up on this position here. We were able to confirm that this is indeed what happens by trapping the intermediate and so isolating this conjugation, this conjugate here, and characterizing it by NMR. So then we went on to use some peptides, as you can see here, peptides with different functionalities next to cysteine. So these were fully shown to be fully cysteine selective. We went on to use some alpha bodies within a site specifically located cysteine. You can see here that we can use a simple building block or a more advanced one where we have a short PEC chain and then a fluorizer that can be inserted in one go onto the alpha body. Yeah. We also did some conjugation with a more with a real protein, let's say, such as bovine serum albumin. And finally, we also did a model conjugation with trastuzumab, proving that we could actually achieve nice conjugation also with these 5-HB2O building blocks. We then determined went on to determine the stability of these conjugates towards thiol exchange. And as you can see here, at pH 7.5, after 10 days, we still have around 80% of the conjugate present. It depends a little bit on the building block, and you can see here that depending on the pH, we can have a fully stable conjugate here illustrated with the carboxylic acid derivative of that 5-HB2O. We could actually completely exclude thiol exchange by doing an additional sodium borohydride reduction on the conjugate, as you can see here. The non-reduced conjugate can still lose a little bit of payload, while if we do a reduction, we can have 100% of intact conjugate after quite a long time, let's say. When we look at these building blocks, they're not only similar to maleimides for conjugation, better in stability, but they also offer the additional advantage that you have in principle, two property-enhancing moieties that can be present on the same building block. And so we generated a series of monofunctionalized 5-HB2Os, where as R1, we have an innocent methyl substituent, which allows us to, as a starting material, to just start from methyl furan. And in our versatile photocatalytic process, we can insert different amides. And you can see that this leads to the decoration of the building block with different moieties, as you can see here. Then, when we vary the substituent on R1 and start from a bit more complicated furan derivatives, you can see here that a versatile array of bifunctionalized derivatives can be generated, where we can, for example, combine a fluorizer with a PEC chain or a long alkyl chain with a PEC chain, as you can see here. So just to give you an idea of the array of protein modification building blocks that we have generated, you can see several examples here. So you can see one that is that introduces a clickable moiety on the amine. Then this one here, which can be transformed into this cross-linker. And I will show you immediately in which scenario this can be used. Then you see again the fluorescent derivative here. You see a combination of, for example, a toxic payload and a long alkyl chain. I want to show you now a bit of a case study where we use these building blocks for conjugation of monoclonal antibodies or proteins in general with oligonucleotides. And so you can see here that to do that, we generate a 5-HB2O derivative of a DNA sequence and then additionally conjugate it with a protein containing a thiol functionality. So let's look a little bit in detail to that scenario. For that purpose, we generate this bifunctional crosslinker here, which contains a 5-HB2O moiety on the one hand for conjugation to a thiol-containing protein, and an NHS ester, an hydroxysuccinamide ester, for conjugation to an amide-containing oligonucleotide. And we use this bifunctional crosslinker in direct comparison with another commercially available one, which contains maleimide and NHS esters. So we took a 5' amino modified DNA sequence and treated that with the NHS ester bifunctional crosslinkers. On the one hand, the maleimide, on the other hand, the 5-HB2O, to generate the resulting adduct, where the R is then supposed to be still a maleimide or a 5-HB2O. And you can see here the crude chromatograms of the reaction. So the green species shows you the 5-HB2O modified DNA, while the gray species here, or the gray rectangle, shows you the maleimide modified sequence. What you can see in this chromatogram is that while the maleimide conjugate can be generated, you see that quickly some products of hydrolysis and other reactions start appearing here. While in the case of the 5-HB2O modified DNA, we just see a little bit of starting material, and apart from that, nothing else generated. We then did a stability test on these conjugates, and so you can see here that for the 5-HB2O conjugated amino DNA, no reaction occurs, and we still keep our conjugation moiety onto that DNA. Comparing that directly with the maleimide, you can see here that this quickly loses its conjugation capacity because it is hydrolyzed depending on the pH quite fast. As you can see here, if you work at pH 9, you can see that it drops down very, very quickly. While the black curve shows you that for what concerns the 5-HB2O, you keep your conjugation moiety. We then went on to do a protein conjugation, and you can see here a model reaction of the 5-HB2O conjugated DNA with an alpha body protein. You can see here how the 5-HB2O DNA in combination with the protein leads to the conjugate that can be isolated here. And you can see here some information about the yield of reaction. So the 5-HB2O reacts slower than the maleimide, as you can see here, but depending on the pH, we can achieve nice yields of conjugates between DNA and, in this case, the alpha body protein. When we look at payload loss, and so again, directly comparing the 5-HB2O oligonucleotide protein conjugate with the maleimide conjugate between a DNA and a protein, we can follow the deconjugation here. So in a huge excess of glutathione, we follow the stability of the conjugate, and we can see for the 5-HB2Os at pH 7 and pH 8 that they're far superior over the maleimide conjugates. With that, I hope to have convinced you. I think my time is probably almost used up. I hope to have convinced you that these building blocks are a nice alternative for maleimides, that we provide hydrolytically stable building blocks, hydrolytically stable conjugates, which are stable towards thiolate chains, and which on top of that have two, what I would call, property enhancing modifications that can be introduced in one go onto a protein. And so I'm referring to the website where you can find the information that Iris Biotech has now nicely provided. And to end, I would like to thank all the collaborators that have actually contributed to this research, You can see quite a few names and collaborators here. I'm not going to name them all in detail. I want to thank my great group of students. That is actually a pleasure to work with. And then you can see some of the funding, and I want to particularly thank the Industrial Research Foundation at UGent for allowing us to try and translate some of our research to real life applications, let's say. And of course, I want to thank you all for your attention.
Yeah, thank you, Annemieke, for sharing those interesting results with us. During your talk, you can imagine we collected some questions. The first is, that was at the very beginning. You said it's proximity sensitive. So what distance is typically the maximum that can be bridged?
Well, that's a nice question. We don't have exact data of distances, but we, in an oligonucleotide context, for example, and maybe I go back to the first slide to show that, if I do that, too many slides, but okay, let me take this one here, for example. Yeah, so in this context, in this case, we have exocyclic amine of the C, which is just in front of the furan. Those T's do not have nucleophilic entities. However, if we replace those T's by C's, then we see that only the one just across engages in a cross-link reaction. On the other hand, so this is an example from the let's say, the nucleic acid context. Also, in the case of proteins, we have been able to show, because there is various lysines present in actin, yeah, we have been able to show that it is the one that is in closest proximity when we look at crystal structure that reacts with the furan. So I cannot really give you distances, but I can give you examples of what works and what doesn't work. So it's a bit of a trial and error scenario. I can also show another example. So this one here, yeah. So for example, if we replace the tryptophan by furan, we get cross-linking. If we replace the tyrosine by furan, still biologically active, no cross-linking. So which means that we have a non-ideal proximity of the target ligand. I hope that answers the question a little bit.
Oh, I absolutely think so, yes. So the next question, I think that came in the context with when you showed that NOx4 producing H2O2. Did you observe off-target activation by reactive oxygen species in living cells?
So in this specific scenario, we only observed oxidation of the furan. So there we do not see off-target oxidation. If we if we go back to the actin-tymosine cross-linking, so let me go back there. Yeah, so here what we do is we use this technology with photosensitizer, irradiation, generation of singlet oxygen. Then in this case, we do see off-target oxidation. So we get some oxidative damage on the other residues in the protein or in the peptide, which is cross-linked. So there we see, for example, of course, methionine oxidizes just by air, so it will also oxidize under these conditions. And we can see, depending on the photosensitizer, the time of irradiation, the concentration of the photosensitizer, we see oxidative damage on histidine and on tyrosine and tryptophan. So we need to carefully fine-tune that. Singlet oxygen is a bullet, and we really need to tame that bullet a little bit. So that is, but on the live cells, we don't see that off-target oxidation. So that's a big advantage of doing it on live cells.
Yeah, definitely. So the next question is, you have described a tautomeric ring closure during conjugation. And yeah, I think you already gave some effects to that. Have you observed payload loss after conjugations? That what then came later? Yeah.
Yeah, that came later. So payload loss after conjugation, we have tested it. I had a slide about that. So here we see that. So after 10 days at pH 737 degrees with 100 equivalents of glutathione, we see some payload loss. However, it is far less than what we observe with maleimides. Yeah,
Definitely,
so if we compare it with maleimides, then we see much less. Also, we can actually completely avoid that payload loss by doing an extra sodium borohydride reduction if the protein is supporting that, of course.
Yeah, sure. Yeah.
Yeah. Okay. So
then a question to the kinetics of the reaction with proteins. How fast is the reaction with proteins? Comparing it to classical maleimides.
The reaction with prolines?
With proteins.
With proteins. Yeah.
Yes.
Okay, yeah, so the reaction, as I said, I mentioned it, is definitely slower. Yeah, maleimides react much faster. This reaction is slower, but if you're interested in a clean conjugate, then this doesn't really represent a real disadvantage because the conjugate is really clean. So you have to wait a little bit longer to get your conjugate. An advantage that we actually observed is that if you have a protein with different cysteines and one is more accessible than the other one, we have examples where maleimides lead to conjugation of both cysteines, whereas the 5-HB2O only goes to the one that is less sterically hindered.
One question that to the solubility, because often when people want to conjugate something to proteins and have something synthetical, which is not very hydrophilic, they have problems to work in aqueous solution. What about the 5-HB2O compared to maleimide constructs? What do you expect with regards to water solubility?
Well, we observe the same problems as with maleimides. So maybe I can go to the cross-linking ones. So let's see this one, for example, where we have an NHS and 5-HB2O. So there are commercially available maleimide derivatives that look the same. So we see that when we see insoluble or solubility problems with maleimides, we of course see them also here, and so that is why we have been studying derivatives where we introduced a polyethylene glycol chain in between the two moieties here. Also, what we have looked at is to use that methyl group here for the introduction, as you can see here, of an extra PEG chain, which then it can actually make the whole building block or crosslinker or whatever more soluble. So we have that extra position. Sorry to interrupt you. We have that extra position.
Because that fits perfectly to the next question. So stay on that slide, I guess. Which functionalities can be incorporated into the building blocks?
Yes. So you see a selection here, and so we have to make a difference. Maybe I can go to the previous slide between the substituents on the nitrogen that we use here and the substituents that actually originate from the original furan. And so if we go back to the synthesis scenario here, this one here, so you can see that actually the substituent, the R1, is there from the start. That is what so you need a furan derivative with a suitable R1, or an R1 that can be modified, and then you can introduce, like, for example, a long alkyl chain or a PEG moiety or so on. This here needs to survive the singlet oxygen conditions. Yeah, so what you have here what you have on the furan and needs to end up at position R1 should not be sensitive to singlet oxygen. Yeah.
Of course, yeah.
Then the next one is the next substituent is actually introduced by the amine. That one is actually exposed to far milder conditions. And so there you have a little bit more possibilities in terms of what you want to introduce on that position. And this is also a different building block. So it depends on building blocks, R1 on Furan, R2 on amines, and then bearing in mind that this first one here has to survive singlet oxygen conditions. Well, the second one is not. So playing with those building blocks and those conditions, we were able to generate, this whole series that I've shown here or on the next slide. So different things that were introduced, and we're still exploring. I mean, if there is a suggestion, we could say, okay, let's see what that gives.
Okay. So well, currently I do not see any further questions within the chat. Again, to all the audience, if you have any questions left, do not hesitate to contact us also afterwards, and we will forward those questions to Annemieke. So
I'll be happy to answer them.
Thank you. Annemieke, thanks again. It was a great pleasure to have you as our guest speaker. With this, then, I would like to close our online workshop. As a follow-up, we will provide the recordings to you. We hope you could collect some helpful ideas and information from this workshop, and you can gain some interesting own experience with this new bioconjugation technique. As you already heard, what a surprise, you can acquire several furan-modified amino acids and the 5-HP2O from today's presentation from Iris Biotech. So please check our web shop or just contact us. Thanks again for joining us. Thanks to all our speakers and also to our QA manager, Klaus Helm, for taking care of the technical support. Enjoy a beautiful Christmas time, stay healthy, and hopefully see you soon. Goodbye.
Bye.
Bye.